Hi everyone, and welcome to Module 8, in which we're going to discuss essential fatty acids and eicosanoids. The objectives of this lecture are to identify and explain the different forms of essential fatty acids in food sources, identify the different forms of eicosanoids, which we'll see are the products of our essential fatty acids, explain two main pathways involved in the biosynthesis of eicosanoids, and explain how incorporating omega-3 into the diet can improve inflammation. Okay, so vast majority of our fatty acids that we require in our diet for optimal biological processes, we can make de novo. So we can make in our bodies, and we talked about fatty acid synthesis in our last module. However, the body is not able to synthesize all of the fatty acid structures that we need. And these are the fats that we can't make are called our essential fatty acids because it's essential that we get these fatty acids from the diet. And these are our omega-6s and our omega-3 polyunsaturated fatty acids. So why do we need these essential fatty acids? Well, they serve two major, to speak in broad terms, two major functions. So first, they are structural components of our cell membranes. And the incorporation of these essential fatty acids into our cell membrane impacts the properties of the cell, including of the membrane, including the fluidity, the flexibility, permeability, signaling, and enzyme activity of the membrane. So you can see that depending on what our diet provides us and what fatty acids we incorporate into our cell membranes, this can have a lot of impact on how our cell functions. Another major function of our essential fatty acids is that they're precursors to lipid biomediators, our eicosanoids, which regulate our cell function. And these eicosanoids are critical in cell-to-cell communication. Okay, so before we talk more about our essential fatty acids, I first want to talk about kind of some nomenclature of polyunsaturated fatty acids. Now, carbon atoms of our PUFAs can be numbered in two different ways. So first, we would have the delta numbering system. And you'll see this little triangle here that's signifying that the author is using this numbering system. And in the delta numbering system, the carboxyl carbon is designated as carbon 1. So we can see our carboxyl group here on our PUFA, and this is carbon 1, and we count from there. We also have our omega numbering system, and this symbol probably looks really familiar because we can talk about omega-3s or omega-6s. And in the omega numbering system, the methyl end is designated as the carbon, so it's opposite of what's shown in this picture. And the number of the bonds from the methyl end indicate the location of the first carbon double bond. So here we have if this is the number 1 carbon, it would go 1, 2, 3, and here's our first double bond, so this would be an omega-3. And here's a couple of examples of how we could use this nomenclature. So we would have, so for our linolenic acid, which is our omega-3, we would have 18, and it would say that this has 3 double bonds, delta, and tells you where all the double bonds are. So this is the delta numbering system. And in the omega numbering system, again, it would be an 18-3, but we would say where the first double bond was. So why are essential fatty acids essential? Well, these are essential in vertebrates because vertebrates lack the enzymes to insert a cis double bond at the omega-6 or omega-3 position of the fatty acid. So in other words, we need to have these essential fatty acids in our body for structure and signaling, but we can't make it because we don't have the enzymes to insert the cis double bond at the specific place where this essential fatty acid needs it to be inserted. So vertebrates are incapable of forming double bonds beyond this delta 9 carbon on the fatty acid chain. And if you flip it, you'll see that the omega 6s or the omega 3s are a delta 12 or a delta 15. However, plants can introduce these double bonds at or plants have enzymes to be able to introduce these double bonds, double double bonds at these higher carbon numbers, and therefore they can synthesize the essential fatty acids. So what these enzymes are called that insert these double bonds is desaturases, and we're going to talk about those a little bit later in the lecture. But these types of desaturases, the omega or the delta 6, 9, 12, or sorry, the 9, 12, and 15, only plants can introduce carbons at those points because only they have the enzymes to do so. And then humans can acquire our essential fatty acids by eating these plants that can make these essential fatty acids. Okay, so let's talk about some of our let's talk about our major forms of omega 6s and omega 3s. So first we have linoleate, which is an 18 carbon PUFA that has two double bonds. And of course, the first one would be at omega 6 for all of these. Next, we have arachidonate, and we'll see that this omega 6 is going to come up over and over again because it has a lot of biological significance. So arachidonate can be synthesized from lineolate, and it's the major substrate for some of our important eicosanoids that we're going to talk about are big mediators in our inflammatory process. So arachidonate can be is a precursor for our eicosanoids called our prostaglandins and leukotrienes. Another omega 6 is this adrenate, which functions as an intercellular storage form of arachidonate. So you can think of it as just a storage form of arachidonate. And then we also have our gamma linolenic acid and dihomo gamma linolenic acid, which are we won't see as commonly in nutrition. Okay, so here are the adequate intake levels for omega 6 fatty acids for various groups. And here are the food sources in which we can find those omega 6s. So it's mostly our seed and bean oils. So we have safflower oil is the highest, then sunflower seeds and oil, pine nuts, sunflower oil, corn oil. So you can see we've got kind of these like vegetable oils, especially this corn oil that we see in high quantities in the Western diet. Then we have our omega 3s, which I think we all know are of high research interest for their biological properties that we'll talk about today. So first we have the alpha linolenic acid, or ALA, and this is the omega 3 that's typically included in infant formulas, but really on a biological level, the main function of ALA is that it can be converted into the other omega 3s, EPA and DHA. Then we have EPA, which is a major component of our tissues, and you can see it's a 20 carbon. So, sorry, I just want to flip back to us for a second and take note that this arachidonate that has such biological significance is also 20 carbons. Okay, so we have our EPA, which is 20 carbons, and then we also have our DHA, which is a 22 carbon omega 3. And DHA is a major component of membrane phospholipids in our eyes, and it's also part of our cerebral gray matter, and it's also has a high concentration in sperm. And here are the adequate intake levels for omega 3s for the different groups. And here are our food sources of ALA. I'm sorry, so this was for, oh yes, this was for all omega-3 fatty acids, but this is food in sources just for the ALA. So we can see we have some oils, seeds, bean oils. And then here are food sources for EPA and DHA. So we can, as we all know, these are in high quantity in different kinds of fish, and one that's not on here is human milk. We also have EPA and DHA in human milk. Okay, and here are the recommended intakes of essential fatty acids according to different countries' recommendations. So here we can see that, so this is from a review article from last year. So we can see that in Canada, um, so sorry, let me back up a little bit. So all of these are recommendations for the ratio of omega-6s to omega-3s that we should get in our diet. And we're going to see why this ratio is important, but in detail. But briefly, omega-6s and omega-3s compete for incorporation into our cell membranes. So basically, I could be eating a lot of omega-3s, but if I'm eating way more omega-6s, then those are what are going to have a higher likelihood of getting into my cell membranes. And my high omega-3 intake isn't going to do a whole lot of good. However, if I have a low omega-6 intake and higher omega-3 intake, then the omega-3s will be more likely to be incorporated into my cell membrane. So more and more, we're starting to make recommendations for our essential fatty acids in terms of the ratio in which we take them in. And in this figure, the authors are showing what the recommendations are for ratios of intake of omega-6s to omega-3 in different countries. So in Canada, they say have a range of either 4 to 1 to 10 to 1. So this is a pretty big range. In the USA, most of our literature says a 2 to 1, 3 to 1, 4 to 1. In France, 5 to 1. In Japan, 2 to 1 to 4 to 1. So you can see there's a lot of variability in what these recommendations are, which I think can speak to the fact that we don't really understand essential fatty acids and the ratio in which we should take them in a lot of detail yet. We don't understand exactly how they work and how they work differently in different physiological settings, such as a disease state. So there's a lot of uncertainty out there, but I think the one thing that we should point out is that in Western diets, which was in the textbook, we have more like a 10 to 1 ratio, and I've even seen as high as a 15 to 1 ratio cited. So that 10 to 1 ratio is at the very limit of some of these recommendations here, and well beyond the limit of many of the other recommendations, including the recommendations for our own country. So generally speaking, Westerners, that is the US, we generally have a much higher intake of these omega-6s, such as from our vegetable oils, compared to omega-3s. So I wanted to touch for a moment on omega-3 supplementation because this is a really hot topic right now. I think there's a lot of ideas out there that these omega-3s are almost a cure-all, that they can address so many health concerns. And that definitely in part is true. But I think I want to talk just briefly about how these omega-3s are found in supplements. So for people who are vegetarian or vegan and want to get their omega-3s through a vegetable source or a plant source, they can get flaxseed oil or linseed oil. Now, like I had discussed before, and we'll talk more about, really the main function of ALA is that it serves as a precursor for EPA and DHA. And I think the general understanding is that there's a pretty low conversion rate of ALA to EPA and DHA. So we really need to take that into account when we're talking with our clients and trying to determine dose. We also see EPA and DHA in supplements, and the content or the quantity of the EPA and DHA and the proportion is really going to vary depending on the specific type of fish oil and how it's been prepared. So we want to encourage our clients to really read the label to try to determine exactly what they are getting in the specific supplement that they've purchased. We also can get DHA supplements from algae or fungal sources, and as well as cod liver oil. But cod liver oil should definitely be used with caution. because it has very high levels of preformed vitamins A and D. So if you're taking high quantities of cod liver oil, you could be overdoing the vitamin A and D. All omega-3 supplements are absorbed more efficiently with a meal. And some people can have kind of unpleasant side effects from the fish oil supplements, especially. They can kind of burp them up or have indigestion or something. And dividing the doses into 2 or 3 smaller doses throughout the day can decrease the risk of side effects. We also may see the omega-6 gamma linoleic acid in supplemental form, and that would be in the form of borage seed oil, evening primrose oil, or black currant seed oil. Now, one pretty recent topic is the of discussion in nutrition is that of the essential fatty acids in infant formula. So we all know from our nutrition training that human milk has more properties that protect infants and their mothers from adverse health conditions. And one of the ideas is that part of this could be because human milk contains essential fatty acids. And those do come from mom, but then they do go into the breast milk. Whereas infant formula traditionally has not included these essential fatty acids. So some formulas have begun to add DHA and arachidonic acid. Remember, arachidonic acid is from the omega 6s to mimic the breast milk. And I just wanted to touch on this because it's just kind of nice to see where current evidence is for these topics that are a little bit more controversial. And I think, as far as I know, the latest evidence has said that the impact on formula supplementation of these essential fatty acids is mixed. So we see some evidence that says it may be beneficial for visual, cognitive, and behavioral development, which we know that our essential fatty acids that we get from the diet and from breast milk do contribute to these outcomes. So it would make sense that supplementing formula would contribute to those outcomes as well. But then some studies show no impact. And again, that kind of also makes sense with what we learn in nutrition, that supplementation just really isn't the same as getting these nutrients directly from the diet. So, in one way, I think when we're talking to clients and making recommendations, we could say that it wouldn't hurt to buy the formula with these essential fatty acids in them. However, we also need to consider that the supplemented formula can be about 15% more expensive than the unsupplemented formulas, and it may not be covered by WIC. I'm not sure what exactly where regulation is now, but last I knew they weren't covered by WIC. So this is a really big consideration for people. 15% more expensive is a lot when you're feeding your baby formula day in and day out every day. And then here is just a little excerpt from a 2014 review article of this that says that while some studies indicate a benefit, others don't. But no matter what, we know that breastfeeding is superior to whatever formula choice you would make, even those that are supplemented with essential fatty acids. But but they do recommend that for people who are formula feeding, that the baby does have some source of DHA and arachidonic acid in the diet. Okay, so as we've discussed, we have two major classes of essential fatty acids. We have our omega 6s and we have our omega 3s. And each have a parent compound that gives rise to longer chain derivatives inside the body. Now, so let's look at this. So here we have our major omega 6 that we find in our diet from our oils. This can be converted to gamma linoleic acid, then dihomogamma linoleic acid, and ultimately the ultimate product of these other omega 6s are arachidonic acid, and this is what has the biological significance. And we can get arachidonic acid directly from the diet in the form of meat, poultry, and eggs. So as we're converting linoleic acid down to arachidonic acid, we are converting it into a longer chain derivative inside the body. So now let's look at our omega 3s. So here at the top we have our ALA, and you see we can get this from many plant sources. Eventually, this is converted to EPA. So remember that this, I believe, is the 18 carbon. The EPA has 20 carbons, and we can get EPA directly from our diet in the form of certain seafoods. And then this is ultimately converted to DHA, which has 22 carbons. So we have more elongation happening um, throughout this process, and our ultimate product is the DHA. Now, there is, as I said, a pretty low efficiency of conversion from ALA down to EPA and DHA. So that's why it's recommended to obtain EPA and DHA from additional sources other than only plant sources, or at least to pay attention to dosage when we're taking in omega 3s only in the form of ALA. Okay. So, how does this elongation occur? So two things are happening in these pathways that we just talked about, and these are a little bit more detailed than in the last figure. But really, as we're going through, as I said, we're elongating our fatty acid chain. So let's just look here. Here we have 18, 18 again, and then we go to 20, and then we go to 22, and then 24 we can ultimately go to. So, and here, let's look at our omega 3 side. We start with 18, and again, we are elongating to 20 carbons and then to 22 carbons. Well, how do we do this? We do this with enzymes that are called elongase enzymes, and that's really the extent of the detail that you need to know for this class. But know that these essential fatty acids are undergoing conversions to longer fatty acids using these elongase enzymes. Now, you can see that we have a different type of enzyme on this pathway as well, and these are these desaturase enzymes. So let's look at the number of double bonds here, okay? So first we have 2, then we go to 3, then we go to 4, and we'll see that when we're going from a 3 double bonds to 4 double bonds, it's through this desaturase enzyme, which makes sense, right? We're desaturating it. We're putting in a carbon double bond so that we have less saturation of hydrogens on there. And it's really, and this is part of the reason why there's competition for incorporation into our membranes between the omega 6s and omega 3s. And that's because these essential fatty acids are competing for desaturase and elongase enzymes in order to get to their more active forms, arachidonic acid, EPA, DHA, that are going to have their biological significance. So that's part of the reason why we have this competition. Okay, now, during an inflammatory response, during an inflammatory response, when we have, our body is going to metabolize arachidonic acid and EPA to form eicosanoids. Now, one take-home message from this is that both arachidonic acid and EPA are both 20 carbon fatty acids. So both of these are incorporated into our cell membranes. And when we have an inflammatory response, our body is going to kind of start breaking down some of our cell membranes in order to, in so doing, it will release these 20 carbon essential fatty acids, and those are going to be metabolized to form eicosanoids. And these are really our biologically, are significant biomediators. So the enzymes that metabolize these 20 carbon fatty acids are called cyclooxygenases and lipooxygenases. Now, as we've talked about in the Western diet, we do get a much higher proportion of omega 6s to omega 3s than in what would be recommended here and throughout the world. So it makes sense that we have more of our omega 6 product, our arachidonic acid, incorporated into our cell membranes. However, when we increase our dietary EPA, we really increase the EPA and decrease the proportion of arachidonic acid to EPA in the cell membrane. And so then we have a higher proportion of, so then when we're going through this inflammatory response and we start breaking down the fatty acids in our cell membranes, if we have had a higher intake of EPA, then we'll have a higher proportion of EPA-derived eicosanoids during this inflammatory response. So our major types of eicosanoids are prostaglandins, thromboxanes, and leukotrienes. So within the cell, arachidonic acid resides predominantly at the C2 position on our membrane phospholipid. Let's think back to the chemistry of the phospholipid, which had two fatty acids on it, and then it had the phosphate group and another nitrogen containing group usually. So within the cell, arachidonic acid is usually at the C2 position of the membrane phospholipid and is released upon activation of phospholipase A2. So we've seen this before. We know that this is the enzyme that releases a lipid from a phospholipid. And in this case, that's going to be arachidonic acid. So if we So I think one thing to remember in this whole conversation is that arachidonic acid is essential too. We also need it for biological functions, including our inflammatory process. Our inflammatory process isn't always a negative thing. We need it in order to protect our body and to fight infection and everything like that. So really, if we didn't get linoleic acid from the diet, which remember is a precursor to arachidonic acid, well, that would threaten our body's ability to synthesize important biomediators as well. So when eicosanoids are formed from the metabolism of arachidonic acid and EPA, they function locally at the site where they were synthesized. And they do this through second messenger systems. Now, I want to take a particular look at what we know about the physiological effects of the eicosanoids prostaglandins. So we know that prostaglandins are natural inflammatory mediators. So in a laboratory model, we can see that administration of certain types of prostaglandins do induce signs of inflammation, including redness and heat, swelling, and edema. And they do these by vasodilation and increasing capillary permeability. Now, we also know that prostaglandins play an important role in reproduction. So we know that, for instance, when used in combination with other drugs, that prostaglandins can be used for termination of pregnancies. We also know that women who have endometriosis have upregulated specific type of prostaglandin, which is what can lead to infertility. We also know that prostaglandins are significant in the process of hypertension, in that certain prostaglandins are vasodilators. So they lower our blood pressure and increase blood flow and decrease peripheral pressure. So we can see that these prostaglandins, depending on what prostaglandin it is, can have different outcomes, but we know that generally speaking, they are heavily involved in the inflammatory response. All right, so let's talk about the ways by which we make eicosanoids from our precursors, arachidonic acid and EPA. So prostaglandins and thromboxanes are synthesized in what we call the cyclic pathway, and leukotrienes are synthesized in what we call the linear pathway. So these are the two pathways by which we make eicosanoids from our essential fatty acids. So in the cyclic pathway it's initiated through the prostaglandin enteral peroxide synthase enzyme, and this enzyme has two activities. First, it has cyclooxygenase and peroxidase enzymes, and It also catalyzes the oxygenation of arachidonic acid together with the cyclicalization of an internal segment of the arachidonic chain. So you don't need to know this in detail, and we're going to have a figure that we'll walk through and look at an overview of it. So it's NSAIDs that, when we have NSAIDs, our non-steroidal anti-inflammatory drugs like ibuprofen and naproxen and aspirin, they are going to work on this cyclooxygenase activity. So here we have the cyclic pathway for the synthesis of clinically relevant prostaglandins and thromboxanes from arachidonic acid. So first we would have any number of stimuli. Here is the bradykinin, but it could also be epinephrine, and that is going to ultimately activate phospholipase A2. We know that phospholipase A2 is going to lyse an arachidonic acid from our cell membrane, right? So here we have that. And then that's going to be metabolized through our cyclooxygenase and peroxidase enzymes in order to form all of these different types of prostaglandins and thromboxanes. Then we have our linear pathway, and this is initiated through the action of the enzyme lipooxygenases. And it's a specific type of lipooxygenase that gives rise to our eicosanoids that are called leukotrienes. And corticosteroid drugs act to inhibit phospholipase 2, therefore inhibiting the release of arachidonate from membrane phospholipids and the subsequent synthesis of the eicosanoids through from arachidonic acid. So here we have, again, a very similar figure where it shows the synthesis of clinically relevant leukotrienes. So again, we could have any numerous stimuli. Here it's bradykinin, but it could also be epinephrine. And again, we're going to activate this phospholipase A2, and then what that's going to do is lyse off arachidonic acid again, and then it will encounter this lipooxygenase enzyme, and then that is how we synthesize our leukotrienes. Okay, so we've talked about that when we have dietary intake of our omega-6s and our omega-3s, they're ultimately converted to arachidonic acid or to EPA, and yes, EPA is further converted to DHA, but and then we know that if this arachidonic acid or this EPA, depending on the proportion which they'll get consumed, they'll either be incorporated more or less into the cell membrane. Then we have some sort of inflammatory stimulus, let's say it's epinephrine or something, that's going to activate phospholipase A2, then that's going to lyse off these fatty acids from our phospholipid membrane, and then they're going to get metabolized into these different kinds of eicosanoids, depending on if it was an arachidonic acid or an EPA that was sliced off. So now we know that EPA has these beneficial health effects. So how does that work? Well, first thing, we know that cyclooxygenase pathway can convert EPA to our a specific type of prostaglandin and thromboxane, and also it can convert it to DHA. We also know that EPA competes with arachidonate for incorporation into the tissue lipids. And we know that when we have more EPA incorporated, then we're going to have less production of eicosanoids from arachidonate. Now, EPA kind of works in two ways you can think of, because in one way, through one mechanism of action, if it's incorporated into the cell membrane, then it's going to inhibit the release of arachidonate and the pro-inflammatory eicosanoids from arachidonate. So that's one way in which it works, by inhibiting arachidonate metabolism. But EPA eicosanoids are also thought to have be anti-inflammatory in and of themselves. So you kind of get a double benefit here. So even though we don't have that high of quantities of EPA in our diet, and therefore we have not that high of quantities of EPA-derived eicosanoids in our bodies, these small amounts still play a really vital role. So for instance, we know that EPA decreases triglyceride production, and in that it's anti-arthrogenic. So by preventing our excessive production of these eicosanoids from arachidonic acid, EPA is really able to fine-tune intercellular signaling to reduce eicosanoid-mediated responses such as inflammation. Okay, so here's a nice little figure here. So here we have omega-3s on this, the left side, so and omega-6s on the right. And it's kind of just repeating what we had seen in prior figures. We have the desaturation, we have the elongation, but here it's showing what the eicosanoid products are of these different essential fatty acids and why we want to have a higher proportion of omega-3s. So here we see that once we get to EPA and they undergo the cyclic or linear pathways, that EPA ultimately produces eicosanoids that are more anti-inflammatory, while arachidonic acid releases or produces eicosanoids that are more pro-inflammatory. So here we have the 3 and 5 series of the prostaglandins, thromboxanes, and leukotrienes, whereas in the arachidonic acid we have the 2 or 4 series of these eicosanoids. We also see that ultimately EPA is converted to DHA, which has its own anti-inflammatory properties. And whatever a person is exposed to, whatever type of eicosanoids they're exposed to, this is going to have an impact on cardiovascular, neurological, and inflammatory disorders. So it's because of the products of this EPA or of omega-3 fatty acids that ultimately becomes EPA that we see these anti-inflammatory properties. And this is why we consider omega-3s to be anti-arthrogenic. So we know that the eicosanoids produced from omega-3s do interfere with platelet aggregation. We know that they are associated with a decreased risk of pro-inflammatory cytokines from cells that are involved in fat plaque formation. And we also know that omega-3s are associated with a reduction in triglyceride concentration. So I kind of went on and on and on about EPA, but we also have specific benefits of DHA. And these, so DHA plays a critical role in the visual and cognitive benefits that we attribute to omega-3s. And it's also implicated in neuronal survival and in signal regulation, including in the brain. And it can even have effects on gene expression in our central nervous system. So what happens if we are these fatty acids are essential, what happens if we have a deficiency? What happens if we don't get them? Well, essential fatty acid deficiency in our population is actually really rare, but it does happen and we do see negative consequences. So if we have a deficiency of omega-6s, this is generally associated with breakdown of our epidermal layer or our skin, and also is associated with water loss because we're having this breakdown of our skin. We attribute dermatitis and for wound healing also to essential fatty acid deficiency. But again, this is incredibly rare. In omega-3s, if we had a deficiency, we would have decreased visual acuity and increased risk of peripheral neuropathy. And that makes sense when we look at the functions of DHA. Okay, so though EFA deficiency is really rare. We historically have seen it in a few different circumstances. So first, we could see it in full-term babies that are fed skim milk, which is low in linoleic acid, and we know for many reasons why we should not feed babies skim milk, but development of essential fatty acid deficiency is one of them. And typically, the negative consequences can be reversed with linoleic acid supplementation. But again, in the United States, these deficiencies are really unlikely to occur because even in formula, infant formula, cow's milk still does have linoleic acid. So infants who are consuming cow's milk will be very unlikely to have an omega-6 deficiency. Now, fat intake in developing countries is low, and much of the fat is from vegetable oils, which we know are really high. Um, fat intake may be low, but the fat that is taken in is usually from vegetable origin, so we know is high in omega 6s. So there won't be linoleic acid deficiency in these circumstances, and there's likely to not be a linoleic acid deficiency either. Now, historically, we have seen essential fatty acid deficiency when patients are undergoing TPN, but now we know to incorporate these essential fatty acids into TPN formulas, so it doesn't really happen anymore. Now, we've talked about why dietary intake of the different essential fatty acids can impact membrane composition. And this really occurs through our entire body except for in our brain. Our brain is the only organ that's resistant to such these like diet induced changes. And you can just think that that really makes sense, that it's a very highly regulated organ and has to have very specific and less mobile properties. But we can do this in many of our other tissues. Incorporate the fats that we eat into our cell membrane so that it affects membrane function. However, in mammals, there are really limits to how much influence we can have on our cell membranes. So our bodies really only allow us to incorporate a certain quantity of unsaturated fatty acids into our diet. So if I have a diet that is, for example, rich in sunflower oils, which are high in linolate, then the omega-6 content of the membrane increases, but that's going to be counterbalanced by a decrease in a different kind of unsaturated fatty acid in the membrane. So yes, we can incorporate more unsaturated fatty acids into our cell membranes, but only to a certain extent because our body wants to protect against excessive membrane fluidity. Now, with all of kind of the rage of omega-3 supplements especially, I think it's pertinent to talk about potential adverse outcomes from overconsumption of omega-3s. So this very, very, very likely will not happen in the diet, but if we're taking these high doses of supplementation, it could. So for our ALA, which remember is from our flaxseed and other plant sources of our omega-3, high doses of flaxseed could cause loose stool or diarrhea, and in certain individuals, they could also have an allergic reaction to flaxseed oil. But other than that, generally no adverse health effects. However, when we go to the more active forms of omega-3s in our bodies, EPA and DHA, we can have negative consequences of overconsumption. And I think that this is being increasingly recognized in the literature. So we know that EPA and DHA affect immune function. And similarly, when we have too much EPA and DHA, they can have a negative consequence on immune function. They also can impact bleeding and increase risk of hemorrhagic stroke and also introduce oxidative damage. And we'll talk about why. Okay, so we talked about that our omega-3 derived, our casinoids derived from omega-3 kind of suppress an inflammatory response, which is generally really good. So it's going to inhibit the eicosanoids, pro-inflammatory eicosanoids from arachidonic acid, and it's going to produce its own anti-inflammatory eicosanoids. And this can be really beneficial for those who have an inflammatory disease or an autoimmune disease where we really want to suppress the immune system. However, in healthy individuals, suppressing the immune system may not always be a good thing because we know that our immune system plays a critical role in protecting our body and destroying pathogens. Now, we also probably know from our education that EPA and DHA can decrease plaque formation and really kind of decrease clotting. They have this kind of anti platelet function, and this can be a really good thing for people who are trying to lower their blood pressure, for instance. But again, for certain individuals, we don't want them to thin out their blood anymore, really, because it can increase risk for bleeding and increase risk for hemorrhagic stroke if supplemented quantities are too high, or if we have a vulnerable individual. And then third, we know that unsaturated fatty acids are vulnerable to lipid peroxidation because they have these double bonds. And this can result in oxidative damage in various tissues. So here we have a nice little figure here where we have a PUFA with a few different double bonds, and then we have a reactive oxygen species that then reacts with this double bond here, and then we have free radicals that are left. So really, by introducing these double bonds, we also are increasing our risk of oxidation to these double bonds. But we can consume more antioxidants such as vitamin E, which is our fat soluble antioxidant, and decrease the risk of this oxidative damage. Okay, and here are your practice questions, and we will talk next week. Thanks, everyone, and have a good week. Bye.